Boy, this is different, the settings. They must have changed something with Zoom. And now it's not working on my other computer. I'm not sure why it's not. All right, share the screen again. In the last slide, all we did was we introduced the operon where it had the promoter, the operon, and then the structural genes in the operon. So the lac operon is the only operon we're going to talk about. It is an inducible operon. So this operon is normally turned off, meaning the lactase enzyme, that gene is normally turned off. And in order to get the enzyme lactase to be made in a bacterial cell like E. coli, the gene has to be turned on. Any questions about that? How the gene is normally turned off is there's a regulatory gene, it's the I gene, and it's not part of the lac operon, it has its own operon. It makes a messenger RNA, which makes a repressor protein, and then that repressor protein binds to the operator on the lac operon. Now, when the RNA polymerase binds to the promoter and then moves down the DNA, it will be stopped by the repressor protein and the operator. And so the lactase gene, the Z or the lac operon, will not be transcribed. Any questions about any of that? And that is normally why the lactase gene is turned off in bacteria such as E. coli. Now, why would E. coli normally want to shut down the lactase gene? That's the Z gene. Anybody have a guess? Well, how often would E. coli see the sugar lactose? Where do we see the sugar lactose? Come on, I know you guys know where you can find the sugar lactose.
DNA?
DNA? No, the sugar lactose is not in DNA. It's in milk and dairy products. And that's about all that you find the sugar lactose. So unless E. coli is on a dairy farm, E. coli is not normally going to see the sugar lactose. And that's why E. coli turns off the lactase gene, because it's not normally going to see the sugar lactose. But when it is around the sugar lactose, you need to have a mechanism for E. coli to turn on the gene. And that happens with an inducer. In the case of the lac operon, the inducer is allolactose, which is an isomer of the sugar lactose. So lactose is, I don't know if it's the left hand isomer, and allolactose is the right hand isomer, or maybe I've got them reversed, it doesn't matter. The point is allolactose is an isomer of the sugar lactose. And whenever you have the sugar lactose around, you'll have allolactose around. And allolactose will bind to the repressor protein and inactivate it. And now the inactive repressor protein cannot bind to the operon of the lac operon. And then the gene gets turned on. Now, let me state that the binding of the repressor protein is not permanent. It's a stoichiomic reaction, and it falls off every once in a while. As long as there's other active repressor protein around, it'll bind to the operator and keep the lac gene turned off. But if the sugar lactose is around, then we'll have allolactose, and it will bind to the repressor protein, so that when the repressor protein comes off of the operon, it will bind to allolactose and then be inactivated, and that will turn on the gene. And let me say that the RNA polymerase, when it binds to the promoter and then moves to the operator, it's not permanently blocked. That the RNA polymerase binds to the DNA with a stoichiomic reaction, and every once in a while it'll fall off the DNA. So it's not permanently blocked in this position when the repressor protein is binding to the operator. It'll eventually fall off and then bind to another promoter and then transcribe that gene. And yes, it could bind to the lac operon promoter and then get blocked by the operator again because of the repressor protein. But in this case, when the RNA polymerase binds to the promoter, it'll move through the operator and transcribe the Z gene, making the messenger RNA for the lactase gene, and then the lactase protein, the enzyme, will be made. And another name for lactase is beta galactosidase. So that's the Z gene. You note that this messenger RNA is a little different. We've never seen this where one messenger RNA is translated into 3 different proteins. In the case of the lac operon, that is the case. Now, in a eukaryote operon, you only have one structural gene in the operon. So only one structural gene will be translated into protein in the operon. But in a prokaryote, there can be more than one gene in the operon. And in the case of the lac operon, there are 3 genes in the structural genes in the operon. Any question about any of that? All right. What more do I need to talk about? It makes sense for E. coli to have an inducible gene, because if the sugar lactose is not around, and that will be most of the time for E. coli, because you don't see the sugar lactose except in dairy products. So unless the E. coli is in a dairy product or on a dairy farm, E. coli is not normally going to see the sugar lactose. And it makes sense to shut it down, because why do you want to make an enzyme if you're not going to need it? If the sugar lactose isn't around, it makes sense to shut down the gene, and then you don't spend any energy making an enzyme that you're not going to be using. But you do need to have a mechanism for turning it on, and that's what the inducer does when the enzyme lactose is around. Any question about any of that? All right, let's move on. Mutation is a change in the genetic material. Mutations can be neutral, where they give no benefit or detriment to the individual who has the mutation, which for eye color, that appears to be a neutral human mutation at the moment. The mutation could be beneficial, where the individual gets a benefit out of the mutation, or it could be a harmful mutation. Most mutations are either harmful or neutral. A mutagen is an agent that increases the frequency of a mutation. You always have a background rate of mutation, and that we call spontaneous mutation. It always occurs in the absence of a mutagen, and that is when the DNA is replicating itself, it makes mistakes every once in a while, not very often, but every once in a while. And that we call a spontaneous mutation. A mutagen increases the rate of a mutation. There are different types of mutations. Here we're actually looking at one that they were looking at, and the boy was, how do you say that? Metallic objects like spoon stuck to him. And they were wondering if the boy had a mutation, and they were looking into it. And, you know, I never heard what the finding was, meaning I heard that they were looking into it, but I never heard what it was. Some people were saying he's magnetic, so he's a mutation. And then other people were saying, no, maybe he just has sticky skin and the spoons are sticking because of his sticky skin. And like I said, I don't know the story about that. But there are different types of mutations. There's a base substitution, also called the point mutation, where one nucleotide is changed for another nucleotide. And there can be different types of base mutations. We'll talk about them. There can be a silent mutation, a missense mutation, and a nonsense mutation. They are all point mutations where one base is substituted for another. There are also frame shift mutations where the entire codon reading frame gets switched. And there are macro mutations where an entire region of a chromosome is added or deleted. And let me warn you that a macro mutation is not discussed in the book, but it is a mutation, so I want to discuss it. It's where the whole region of DNA is changed. And actually, you know of a macromutation. Down syndrome is where a child is born with an entire extra chromosome. They have 3 copies, I think it's chromosome 21, and that's a macromutation. A base substitution is where the wrong nucleotide gets inserted. So instead of C going there, T is put in there. And then when this gets replicated, the A will be put in. And now this mutation is fixed because there's a T and an A in there. In this case, when the T is put in there, the cell can notice that this DNA is a mismatch there, and it can remove that T and then put in a C and correct the mutation. But at this point, that mutation is now fixed. Okay, any question about that? That's a spontaneous mutation where the DNA polymerase just put in the wrong base. And that is a base substitution point mutation. Let's talk about different types. There's a silent mutation where a nucleotide in the DNA gets changed, but the nucleotide in the DNA still codes for the same amino acid, like U codes for phenylalanine, and a silent mutation would be when this third U is replaced with a C. Well, the protein will not be changed, so this we call a silent mutation. It has no effect on the protein function. Any question about that? A missense mutation is when you change one base and it results in a different codon that codes for a different amino acid. So that in the protein, one of the amino acids will be changed. An example of the missense mutation is sickle cell anemia, where in a protein, hemoglobin, containing thousands of amino acids, only one of them is changed. And yet that protein does not work well. It does function, but it doesn't function well. And the patient, if it has two of the genes for sickle cell anemia, will have sickle cell anemia disease. Any question about any of that? Another example of a missense mutation that happened in the human population about 10,000 years ago was the brown eye gene was changed into the blue eye gene. And that happened about 10,000 years ago. And it's true, you need two of those genes to get the expression of the blue eye. But when the first mutation happened, it was passed on, and probably because of the low population inbreeding happened. And then a child was born with two of the blue eye genes, and then the child had blue eyes. As I said, that happened in Northern Europe about 10,000 years ago. Any question about any of that? All right, can somebody confirm that you guys are hearing me? Hello?
Yeah, I can hear you just fine.
Yes.
Thank you. All right. That's today, the missense mutation for the blue eye gene appears to be neutral today, but in the past it may have been selected for, because in Northern Europe, the blue-eyed allele, the gene, is more common than actually the brown-eyed allele. And initially, everybody in the human race had the brown-eyed allele. So initially, it probably was selected for, but today it's now a neutral mutation. Another mutation we need to talk about is a nonsense mutation. This is where you change one nucleotide base in the DNA, and it results in a stop codon in the, the codon of the messenger RNA or the protein. And that means that you get, instead of the normal protein, you get a truncated protein. And this is called a nonsense mutation, where when that mutation occurs, the rest of the gene is not translated, and so you got a shorter protein. Oftentimes, the protein will not work if you have a nonsense mutation, but in a few cases, the protein works. It just doesn't work optimally. Some examples of a nonsense mutation are some muscular dystrophy and some cystic fibrosis patients. All right. The next mutation we want to talk about is a frame shift mutation. That is where you insert one or more nucleotides into the DNA, or you delete one or more nucleotides from the DNA. So here we have UUU coding for the amino acid phenylalanine, and we remove one U or one A on the other base. And then the codon, instead of being UUA for phenylalanine, will now grab the next nucleotide, which is a G, and so it'll be UUG, which will switch the amino acid from phenylalanine to leucine. But take a look now at the next codon. It used to be GGC, but that G has moved up now into the UUG codon. So the new codon will be GCU, coding for another amino acid. And that will shift all of the codons. Everybody see that? That's why it's called a frame shift mutation. When you add or remove one nucleotide, this codon is changed, but all subsequent codons are changed. And that happens whenever you insert or delete any number of codons, nucleotide bases, except 3. If you were to remove UUU3, it's not a frame shift mutation. It's a, what is that, a missense mutation? Because you're removing one amino acid, but all the other amino acids in the protein are present. All right, any question about that? So a multiple of 3, either deletions or addition, is not a frame shift mutation. But an insertion or deletion of 1 or 2 or any number of greater than that that's not divisible by 3 is a frame shift mutation. And some Tay-Sachs patients are frame shift mutations. Okay, so when we're talking about mutations, you should realize that mutations can be caused by one of 2 events. They can be spontaneous mutations, where the DNA just is mutated at the spontaneous background rate because the DNA nucleotide made a mistake when it copied the DNA. Mutations can also happen when a mutagen is present, and that increases the rate of the mutation. And there's 2 types of mutagens. There's radiation and there's chemical mutagens. Radiation, there's 2 types, ionizing radiation and non-ionizing radiation. Ionizing radiation has a large amount of energy, like X-rays or gamma rays. And when the ionizing radiation hits an molecule or an atom, it gives the molecule or atom so much energy that that electron takes the energy and then leaves the atom, creating a atom or molecule behind that becomes ionized because it gets a +1 charge because it's lost 1 electron. These ions can then react to DNA, increasing the rate of a mutation. So that's why you shouldn't be exposed to ionizing radiation like X-rays, because they'll hit a molecule, creating an ion, and then the ion can damage the DNA. Any question about ionizing radiation? If not, let's talk about non-ionizing radiation. This is from UV light. UV radiation is the only non-ionizing radiation that you need to worry about. UV light, when it hits DNA, will cause the DNA to react where if the DNA has two thymines that are next to each other, the thymines absorb the UV light, and then that forms a thymine dimer where the two thymines actually form a covalent bond between them. And DNA nucleotides do not normally bind to each other. The thymine dimer creates a problem because once the thymines start binding to each other, they will not hydrogen bond with their corresponding nucleotide, the A's. And this will cause a bubble in the DNA because this is incorrect. The thymines are binding to each other with the dimer, two bonds, covalent bonds. They're not binding to the A. This region of the DNA cannot be replicated or it cannot be transcribed. Any question about any of that? Anyways, that's how UV radiation causes mutations, by causing the thymines to form two bonds to each other. We call that a thymine dimer. We're not going to talk about how you can repair that. Let's talk briefly about chemical mutagens. We're only going to discuss it with nitrous acid, which is a chemical mutagen. When nitrous acid gets in a cell, it can react to the DNA and then change the nucleotide. This A here gets modified, and it's still an A, but it's a modified A because it's been exposed to nitrous acid. And now this modified A, when the DNA gets replicated, the modified A, instead of binding to a T, it's going to bind to a C. So this modified A is going to look to the DNA polymerase like a G, and then it'll insert a C here. And then when this gets replicated, we'll have, instead of an AT here, we'll have a CG. And then this cannot be repaired. Okay, at this point, although this would be hard to repair because it's going to see that A as a G, but it could always replace that C and then put in the T, but I'm not sure how it could do that because the modified A looks like a G. But anyways, once the DNA gets replicated, the mutation is now fixed. Any question about any of that? All right, so let's talk about the frequency of mutation. The spontaneous mutation rate is that every 1 times 10 to the 9th replicated bases, the DNA polymerase will make a mistake and put in the wrong nucleotide. So the frequency of a spontaneous mutation for a nucleotide is 1 in 10 to the 9th bases. Now, a gene has about 1,000 nucleotides. So that means the mutation rate for a gene is 1 in 10 to the 6th, or the frequency of a mutation for a gene is that one gene will be mutated out of 1,000,000 genes. Now, that's not very much, is it? However, if we look at the frequency of the human population, it's over 1 million. In fact, it's, what is it? Over 7 billion, almost 8 billion, not 8 billion. Yeah. Yeah, 8 billion, almost 8 billion. So in the human population, every once in a while, we'll get a child born who has a spontaneous mutation in a gene. Any question about that? All right. Mutagens increase the mutation rate by 10 to 1,000 times. So instead of one gene in 10 to the 6 being mutated, it'll be one gene in 1,000 or one gene in 10 to the 5th genes that are mutated. Any question about any of that? Well, you can identify mutants experimentally. Like if you were to see somebody walking down the street like that, you'd say, yep, there's a mutation. And that would be a positive direct selection where you can detect the mutant because they grow or appear different. Like the first time somebody was born with the blue-eyed gene, you could see that difference, and that would be a positive direct selection where you could detect the mutant cell. A negative or indirect selection, you can detect the mutant cells because they do not grow. And I'll give you an example of that in a minute. In the next slide, actually. So here we have a plate having colonies, and you can get a velvet surface and stamp the colonies, and then stamp the velvet onto an agar plate. And then you'll get the colonies growing in the same position they were in your initial plate having the colonies. Well, if you were to stamp a plate containing the amino acid histidine and a plate lacking the amino acid histidine, if this colony here, which is this colony here, and that colony there, if it would not grow on the plate lacking the amino acid histidine, but it would grow on a plate containing the amino acid histidine, you could say that this colony is a mutant, and this colony cannot make the amino acid histidine, in which case it will not grow on a plate lacking that amino acid. This is negative selection for mutations, where we're identifying mutation because it's not growing. Any question about any of that? All right, if not, let's move on. Genetic recombination is the exchange of genes between two DNA molecules. Recombination is similar to crossing over. Crossing over is when two DNA molecules come together and then literally cross over, and then they break right here, where one portion of the chromosome, let's say that's the mother's chromosome, combines with the father's chromosome that came from the father. And then we get a mixed chromosome, where the lowercase alleles in gray are the chromosome that came from the mother, and then the portion of the blue chromosome in the capital letters, this part of the chromosome came from the father. This is crossing over. Recombination and crossing over can be the same, meaning whenever you have crossing over occurring, that leads to recombination, an exchange of genes between two DNA molecules. However, recombination is a little more general than crossing over. And you can have recombination occurring without crossing over occurring. For example, a recombination that's happening and crossing over is not happening when a DNA molecule is pulled into the cell, taken up by the cell, and then that DNA molecule gets inserted into the chromosome, and usually a region of the DNA is lost from that chromosome during that insertion. This will be a case of recombination that happened without crossing over. Any question about any of that? So recombination is a little more general than crossing over, but oftentimes they do mean the same thing. Recombination doesn't add new genes to an individual or even to the population. However, it reassorts the genes. For example, we have all of the capital alleles, like capital H, capital I, with the capital G. Well, after crossing over, the capital H will now be with the lowercase g, and that shuffles the DNA in a population. Any question about any of that? And when you shuffle the DNA in the population, you can have new combination of genes, like the lowercase g is now with the uppercase H. And previously that wasn't the case. For example, in Northern Europe, a case of recombination was the blue-eyed allele was on the same chromosome as the gene for blonde hair. And over time, the blue-eyed allele recombined, and now the blue-eyed allele can be on the chromosome for brown hair or any other color of hair. It's not always with blonde hair, and that's because of recombination, shuffling the gene combinations in the population. Any question about any of that? All right. Genetic transfer is where DNA from one cell leaves that cell and then gets taken up by another cell. And you can have genetic transfer occurring in two different ways. There's vertical gene transfer. This occurs during reproduction between generations of cells where the parent cell transfers its DNA to the offspring cell. For example, the mother transfers half of her genes to her offspring. Vertical gene transfer. There's also horizontal gene transfer, where the genes between cells of the same generation move around, where a donor cell gives up the DNA, probably when it dies, and then that DNA is taken up by a recipient cell. There are three ways that horizontal gene transfer can happen: by transformation, conjugation, and transduction. Transformation is where DNA comes out of a cell. Usually that DNA comes out of a dead cell. And then this DNA is taken up by a recipient cell. Let me blow this up a little. And then this DNA can recombine with the portion of the chromosome, which normally the chromosome had a capital A, and then that recombines with the DNA that came into the cell. And now the capital A is removed from the chromosome, and the lowercase a came into the cell. And the lowercase a came into the DNA, was from the DNA that got taken up into the cell. This is transformation. Any questions about transformation? If not, let me state that transformation in bacteria happens about 1 in every 10 to the 5th genes in bacteria. Or transformation happens 10 times more common than spontaneous mutation. Transformation is a way for this cell to get a new gene, like the lowercase a. Maybe that gave tetracycline resistance. And now the lowercase a has been taken up by the recipient cell, and this cell will now be resistant to tetracycline, whereas before it was sensitive. And here we're looking at a cell taking up DNA, and then it expresses that DNA, and its phenotype changed from transformation. Any questions? The second way that new DNA can be taken up by a cell is by conjugation, also called sexual conjugation. In gram negatives, sexual conjugation happens with the sex pilus, where there's an F plus cell that makes a sex pilus, and that sex pilus literally skewers an F minus cell and then brings the two cells closer together, acting like a winch. The cells do not have to come right next to each other, meaning that the sex pilus is hollow and a plasmid can move from the F plus cell to the F minus cell in sexual conjugation. That's the way gram negatives do sexual conjugation. Gram positives do sexual conjugation in a different manner, and that is the two cells come together and then they fuse their cell membrane so that this cell membrane of this cell now connects to the cell membrane of this cell. And then the cell membrane goes around and now connects to the cell membrane of that cell, meaning that the cytoplasm of these two cells are now interconnected. And a plasmid or a piece of DNA can move out of this cell and move into that cell. And this we call a mating bridge. And you can find mating bridges in gram-positive cells. Any question about any of this? In conjugation, the sex pilus skewers the F minus cell and then brings them together, although the cells didn't have to be this close. The sex pilus is hollow. And then this F plasmid makes a copy of itself and then sends a copy through the sex pilus to the F minus cell. The F minus cell now has the F plasmid. The F plasmid has the genes for making a sex pilus. And so this F minus cell becomes converted into an F plus cell. And both of these cells are now F plus cells. Okay. And then this cell that used to be the F minus will grow a sex pilus. And then that sex pilus can skewer an F minus cell and then go through sexual conjugation again. Any questions about sexual conjugation? The third way that horizontal gene transfer can happen uses a virus in a process that we're going to call transduction. Normally, when a virus infects a cell, it will pump its DNA into the infected cell, and then the DNA wants to run the show in the cell, and the first thing it'll do is degrade the host chromosome. Why? Because if you remove the host chromosome, then only your DNA is in the cell to run the cell. And then the virus will replicate its DNA, transcribe, so there's more copies of the viral DNA, and then transcribe the viral DNA to make viral proteins. And that will make the head of the virus the capsid. I know we haven't talked about viruses yet, but we will. And then the viral DNA, which is pinkish, gets added to the protein coat, the head of the virus. And then the virus can lyse the cell, releasing the virus, and then the virus can go out and infect another cell. Any question about any of that? All right. Well, probably because viruses make lots of offspring, we don't worry about if they make a mistake. And as you can see in this case here, this virus did not get the red virus DNA added to the virus. What happened was there was a piece of the host chromosome, and that's shown in purple, got added to that virus instead of the replicated viral DNA, which is pink, which was not added to this. Now let's watch what happens when this virus infects another cell. It infects the cell and injects its DNA into the cell, but this is not viral DNA. This is DNA from the host chromosome of this cell. And then that host chromosome DNA can recombine with the chromosome, and then the new cell, the cell can get a new gene which can be expressed, and this we call the recombinant cell. And this new DNA can change the properties of this cell. For example, if this gene coded for, let's go ampicillin resistance, and that DNA was inserted into this virus, which inserts that DNA into this cell. There's the DNA that came from this cell, and it will carry the ampicillin resistance to this cell. So now this cell is ampicillin resistant, whereas before it was sensitive to that amino, to ampicillin, the antibiotic ampicillin. Having trouble with that word for some reason. So you can see through transduction that this cell can pick up a new gene that it previously did not have. And how often does transduction occur, as well as conjugation? All of them, including transformation, all of them happen in one cell in 10 to the 5th, meaning that if a cell picks up a new gene, it probably picked it up by transduction, conjugation, or transformation, rather than picking it up by spontaneous mutation. Any question about that? All right, we need to briefly talk about plasmids. Plasmids are a small piece of DNA which is not contained in the chromosome. It is similar to the chromosome, other than its size, that it's circular DNA. It does contain genes, but generally it contains only a handful of genes, whereas the chromosome can contain tens or hundreds of thousands of genes. Why plasmids are important is that because they replicate independently of the cell's chromosome, the plasmid can have more than one copy in the cell. And then when the two cells come together, like in conjugation, the plasmid can move from one cell into the other. So plasmids allow DNA to more easily move between cells of the same generation. And that will help shuffle the genes within a population, mix up the genes. Any question about any of that? Now let me state that there are different categories of plasmids. All you need to know is that they are different categories of plasmids. You don't need to know the different types of categories. Another way that genes can move around in a cell or in a population is by a transposon. Transposons are segments of DNA that move from one region of DNA to another. For example, a transposon in human DNA on the X chromosome could leave the X chromosome and then move to the first chromosome. Any questions about any of that? So transposons are pieces of DNA that move from one region of the DNA to another. Transposons always encode the transposase gene, and that literally cuts out the transposon here and here, and then binds the DNA together, meaning this portion of the DNA will be bound to this portion of the DNA. The transposase gene does that. And then the transposase gene cuts the DNA and then allows the transposon to bind to where that DNA is cut. And then the transposase gene links the DNA together. Transposons always contain the transposase gene, but they also contain inverted repeat, where the nucleotides are inverted and repeated on both ends of the transposon. Complex transposons can carry other genes in the transposon, such as this transposon here, which includes the gene for kanamycin resistance. Now, transposons can move from the chromosome into a plasmid, and then the plasmid would be more likely to move from one cell to another cell, moving around the DNA, not only in the genome of the cell, but between different cells of a population. And that's important because kanamycin resistance can move around in the cell from one location to another. and then can move into a plasmid, like here. And then this plasmid can move into another cell, spreading around kanamycin resistance. Any questions about transposons? All right, so the last topic we need to talk about are genes and evolution. All new genetic variation that arises in a population has to come from mutation, meaning if the genetic variation for that gene did not exist previously, then the only way it can enter the population is through spontaneous mutation. And I already gave you an example. 10,000 years ago, humans only had the gene for brown or brownish eyes. But 10,000 years ago, through spontaneous mutation, humans obtained the blue-eyed gene through a mutation. Crossing over is important in the genetic variation of the species. It doesn't add new genetic variation, but it puts new combination of genes together. Transformation, conjugation, and transduction are a way for a cell to obtain new genetic variation. But this new genetic variation has to exist in the population. And if it exists, it can move from one cell to another by transformation, conjugation, or transduction. And so a new cell can pick up a new gene as long as the gene exists in the population. If it doesn't exist, then the only way a cell can pick up the new genetic variation is through spontaneous mutation. Transposition is similar to crossing over in that it helps new combination of genes come together. All of these can shuffle the genetic variation in a population, and then evolution can happen in a population as natural selection selects for the genetic variants that have the greatest reproductive effect. Meaning if a gene is beneficial to the individual in the species, in this population, that will be selected for, and that population will increase in that genetic variation. And that is evolution. Any question about any of that? If not, let's just start. Oh, I don't have it here. Let's start chapter 14. And I might need my thumb drive. Because I transferred chapter 14 to the computer, so it's not here. Can you guys hear me? I'm not using the headphone at the moment.
Yeah.
Thank you. All right. Let me remove this. Put my headphone back. Slowed down by a lot of things today. Chapter 14, the principles of disease and epidemiology. This slide is introducing the major goals of this chapter and the rough outline. You should know the terms of this chapter. And in this chapter, if you know the terms that are introduced to this chapter, you're going to learn about 50% of the material for this chapter, meaning terms are very important to chapter 14. Understand the relationship between the normal microbiota and its host. Understand the different stages of disease development, and understand about nosocomial infections and how to control them. Let me mention that if you guys are going into the healthcare field, you should learn about nosocomial infections and how to prevent them, because this is a serious disease. And this will be one of the greatest things you can learn in microbiology if you're going to work in the healthcare facilities, healthcare field. Any question about any of that?
What are they doing to that guy's back in the picture?
I'm sorry? Can you
What are they
repeat
doing to that guy's back in the picture?
that? You're not seeing the picture?
No, but what are they doing to his back with the?
I don't know. It looks like they're suctioning his back. This is a long time ago, one of the early physicians, and it looks like he's got a lamp there, so he might be going to be sterilizing his instruments, and he might be doing surgery on the boy. I don't know. Okay, but I just thought it was a cool picture, so I inserted it. And it is talking about disease and epidemiology. It's giving you an example of that. So know that pathology is the study of disease, etiology, the study of the cause of a disease, like the etiological agent of anthrax is Bacillus anthracis. You should know the term pathogenesis, the development of a disease. Infection is colonization of the body by a pathogen or the presence of a microbe where it is not normally found. So if your body gets colonized by a pathogen, you can have an infection, which can then damage the body. And you can get an infection where by a microbe that's on your body, but it's not supposed to be found there, like Staphylococcus epidermidis is normally found on the skin and it doesn't do any harm to the skin. But if it's growing internally, like in a puncture wound, that would cause an infection. So presence of the microbe where it's not normally found. A disease is an abnormal state in which the body is not functioning normally. And disease can be caused by an infection, but it can happen like with old age, for example, like tennis elbow is not being caused by a pathogen. You should understand that some microorganisms are pathogenic, meaning they cause disease, but few microorganisms are pathogenic. Most microorganisms are not pathogenic. Some microorganisms even benefit the host. For example, all of the microorganisms, I shouldn't say all of the many of the microorganisms that form the normal microbiota of the host are beneficial. Not all, but many of them are beneficial to the host. The normal microbiota establishes a relationship with its host. The normal microbiota is a large community of microbes found in association with all animals and plants. And this we call the normal microbiota, where the microbes tend to permanently colonize and then live on the host. For example, a child who's born would initially not have Staphylococcus epidermidis on their skin, but as soon as their skin touches the mother's skin during birth, the child will pick up Staphylococcus epidermidis as part of their normal microbiota. And generally, the child will have Staphylococcus epidermidis for their lifetime. The normal microbiota, such as Staphylococcus epidermidis, is only found in certain regions of the body, such as the skin in this case. If it's found in other regions of the body, it's not supposed to be there and it can cause disease, such as in a puncture wound in the internal tissues. I don't know if Staphylococcus epidermidis can cause pneumonia, but if it were to grow in the lungs, it would definitely cause pneumonia. You can disrupt the normal microbiota host balance, and that appears to be associated with disease and impaired immune function. For example, we all have E. coli, large numbers in our gut, and whenever you take an antibiotic, assuming that antibiotic works on gram negatives, that will knock down the E. coli in your gut. That would disrupt the normal microbiota host balance. That disruption of the host balance, the normal microbiota host balance, could lead to diarrhea or constipation in the patient. And that is something that we frequently see when we put a patient on antibiotics, where they may develop diarrhea or constipation simply because the antibiotic disrupt the normal microbiota host balance. Fortunately, usually the balance comes back into maybe not the same original balance, but into a new balance. And then the patient will get better, where their diarrhea will go away or their constipation will go away. Any questions about any of that? All right, so this slide is trying to show you the locations of the normal microbiota in the human body.
It's 6:20.
Yeah, I know. Let me just finish this slide.
Okay.
The skin, the eyes, the mouth, the nose, the throat, the gastrointestinal system, and then the lower urinary and reproductive systems, meaning the urethra in both males and females is colonized by the normal microbiota. The normal microbiota is not normally found internally, such as the blood, the cerebrospinal fluid, and the bones. A transient microbiota is similar to the normal microbiota, but the transient microbiota may be present for days, weeks, or months, where an organism colonizes a patient or a host. But unlike the normal microbiota, the organism goes away. It's transient. For example, I think I may have mentioned this before. I hugged my, well, actually she hugged me, my aunt, when she had a Staphylococcus aureus infection. And after I hugged her, I noticed I had Staphylococcus aureus on my skin. And I said, geez, hugging my aunt got me Staphylococcus aureus. I hope that goes away. And 3 months later, I tested my skin and I did not have Staphylococcus aureus anymore. So that was for me a transient microbiota. Any question about any of that? If not, come on. Oh, yeah, I have it here. If not, I will end this here and I'll see you at 6:30 for the lab. Bye.